Steps in the cell cycle. The cell cycle is a series of events that take place in a cell leading to its division and duplication. It is a complex and highly regulated process that ensures proper cell growth, DNA replication, and division. The cell cycle is generally divided into two major phases, interphase and meiotic phase, M phase. Each phase has distinct steps that contribute to the preparation of a cell for division. Below is a detailed explanation of each step in the cell cycle. 1. Interphase. Interphase is the longest phase of the cell cycle, during which the cell grows, performs its normal functions, and prepares for division. It is further divided into three subphases, G1 gap 1 S synthesis and G2 gap 2. A. G1 phase gap 1. Function. The G1 phase is the first stage of interphase and is primarily focused on cell growth and normal cellular functions. During this phase, the cell increases in size, synthesizes proteins, and produces RNA and other molecules necessary for DNA replication. Regulation. At the end of G1, the cell faces the G1 checkpoint, which checks for DNA damage, adequate resources, and favorable environmental conditions for DNA synthesis. If any issues are detected, the cell may enter the G0 phase, where it remains in a quiescent state until conditions improve. Outcome. If everything is in order, the cell proceeds to the S phase. B. S phase, synthesis. Function. The primary event in the S phase is the replication of the cell's DNA. Each chromosome is duplicated to produce two sister chromatids connected by a centromere. Regulation. The initiation of DNA replication is tightly regulated to ensure that it occurs only once per cell cycle. Proteins such as cyclin ECDK2 and cyclin ACDK2 are involved in initiating DNA replication. The S checkpoint ensures that the replication process proceeds accurately. Outcome. At the end of the S phase, the cell has two complete copies of each chromosome, each consisting of two sister chromatids. C. G2 phase gap 2. Function. The G2 phase follows the S phase and is a period of preparation for mitosis. The cell continues to grow and produce proteins required for cell division. Key processes include the synthesis of tubulin, which will be used to form the meiotic spindle. Regulation. The G2 checkpoint ensures that DNA replication has been completed correctly and that there is no DNA damage. If any damage is detected, the cell may halt its progress to allow for repair. Outcome. If the cell passes the G2 checkpoint, it proceeds to the meiotic phase. 2. Meiotic phase, M phase. The meiotic phase is where the cell physically divides into two daughter cells. It consists of mitosis, which is the division of the nucleus, and cytokinesis, the division of the cytoplasm. A. Mitosis. Mitosis is further divided into 5 stages: prophase, metaphase, anaphase, telophase, and cytokinesis. During mitosis, the cell's replicated chromosomes are evenly distributed into two daughter cells. Prophase. The chromosomes condense into distinct structures visible under a microscope. The nuclear envelope begins to break down, and the meiotic spindle begins to form from microtubules. The centrioles in animal cells move toward opposite poles of the cell. Metaphase. The chromosomes align at the metaphase plate, the middle of the cell. Each chromosome is attached to the spindle fibers via its centromere. The spindle checkpoint ensures that all chromosomes are correctly attached to the spindle before the cell proceeds to anaphase. Anaphase. The sister chromatids are pulled apart as the centromere divides, and they move toward opposite poles of the cell. The cohesin proteins that hold the sister chromatids together are cleaved, allowing for their separation. Telophase. The chromatids, now individual chromosomes, reach the opposite poles of the cell. The nuclear envelope begins to reform around each set of chromosomes, creating two new nuclei. The chromosomes begin to decondense back into chromatin. B. Cytokinesis. Cytokinesis is the final step of the M phase and involves the division of the cytoplasm, resulting in two distinct daughter cells. In animal cells, a contractile ring made of actin filaments forms and pinches the cell membrane in two, a process known as cleavage furrow formation. In plant cells, a cell plate forms in the middle of the cell, which eventually develops into the new cell wall that divides the two daughter cells. After cytokinesis, the two daughter cells enter G1 phase of the next cell cycle, and the cycle repeats. Regulation of the cell cycle. The progression of the cell cycle is tightly controlled by various checkpoints, proteins, and enzymes to ensure accurate cell division. These regulatory mechanisms include cyclins and cyclin-dependent kinases, CDKs. Cyclins bind to CDKs to form active complexes that drive the progression of the cell cycle. The levels of cyclins fluctuate throughout the cycle, and their binding to CDKs is critical for the transition from one phase to the next. Cell cycle checkpoints. G1 checkpoint restriction point ensures that the cell is ready for DNA synthesis and checks for DNA damage. G2 checkpoint ensures DNA replication is complete and checks for damage before mitosis. M checkpoint ensures that all chromosomes are properly aligned before anaphase. Tumor suppressors and oncogenes. P53, a key tumor suppressor that monitors DNA integrity and induces cell cycle arrest or apoptosis if DNA damage is detected. RB retinoblastoma protein regulates the transition from G1 to S phase by inhibiting E2F transcription factors. Cyclin D CDK4 promotes the transition from G1 to S phase. APC regulates the separation of sister chromatids during anaphase by targeting specific proteins for degradation. Clinical relevance. The cell cycle's regulation is crucial for maintaining genomic stability. Disruption of this regulation can lead to cancer, where cells divide uncontrollably due to mutations in genes regulating the cycle, such as mutations in P53, RB, or cyclins. Understanding these processes is essential for developing targeted therapies for cancer and other cell cycle-related diseases. Solved previous year question. Q. At which stage of the cell cycle does DNA replication occur? A. G1 phase, B. S phase, C. G2 phase, D. M phase. Answer: B. S phase. Explanation. DNA replication occurs during the S phase of the cell cycle, where each chromosome is duplicated to produce two sister chromatids. MCQs. Which phase of the cell cycle involves the replication of DNA? A. G1 phase, B. S phase, C. G2 phase, D. M phase. What is the primary function of the G1 checkpoint? A. Ensure DNA replication is complete. B. Ensure proper chromosome alignment. C. Ensure cell size and nutrients are adequate for DNA synthesis. D. Trigger cell division. Which of the following proteins is responsible for the degradation of cohesins during anaphase? A. Cyclin BB, APC, CC, securin D, P53. Let me know if you'd like to proceed with any further topics or questions. You said. Regulation and control of cell cycle. ChatGPT said. Regulation and control of the cell cycle. The cell cycle is a highly regulated process, and its control is essential for maintaining proper cell function, division, and overall organismal health. Disruption in the regulation of the cell cycle can lead to uncontrolled cell division, which is a hallmark of cancer. The regulation and control of the cell cycle involve multiple mechanisms, including checkpoints, cyclins, cyclin-dependent kinases, CDKs, tumor suppressors, and various feedback loops. Key regulators of the cell cycle: cyclins and cyclin-dependent kinases, CDKs. The primary regulators of the cell cycle are cyclins and cyclin-dependent kinases, CDKs. Cyclins are proteins whose levels vary throughout the cell cycle, and they bind to CDKs to activate them. The activated cyclin-CDK complexes regulate the progression of the cell cycle by phosphorylating target proteins. Cyclins. These proteins accumulate and degrade in a cyclical manner during the cell cycle. Each cyclin is specific to a particular phase. For example, cyclin D binds with CDK4/6 to promote the transition from G1 to S phase. Cyclin E binds with CDK2 to allow progression into the S phase. Cyclin A binds with CDK2 during the S phase to ensure DNA replication is completed. Cyclin B binds with CDK1 to promote the transition from G2 to M phase. Cyclin-dependent kinases, CDKs. These enzymes are activated by binding to cyclins and are responsible for phosphorylating various target proteins. This phosphorylation is crucial for driving the cell through different stages of the cell cycle. CDKs are active only when associated with their specific cyclins. Cell cycle checkpoints. Checkpoints are control mechanisms that ensure the proper progression of the cell cycle by monitoring the integrity of DNA, spindle attachment, and other key cellular conditions. If any abnormalities are detected, the cell cycle is halted until the issue is resolved. G1 checkpoint, restriction point. This checkpoint occurs at the end of G1 phase, and it is crucial for determining whether the cell is ready to proceed to the S phase and replicate its DNA. The decision to pass through the G1 checkpoint is influenced by external signals, such as growth factors, and internal factors, such as the cell size and nutrient availability. If the DNA is damaged or the cell is not large enough, the checkpoint will halt progression, and the cell can either enter G0 phase, a resting state, or undergo repair mechanisms before proceeding. The RB protein, retinoblastoma protein, plays a key role in this checkpoint by binding to and inhibiting E2F transcription factors, which are required for the transition into the S phase. Phosphorylation of RB by cyclin D CDK4 leads to the release of E2F, allowing DNA replication to proceed. G2 checkpoint. This checkpoint ensures that the cell has successfully completed DNA replication and checks for DNA damage before the cell proceeds to mitosis. If DNA replication is incomplete or if damage is detected, the checkpoint will halt the cycle, allowing time for repair. The ATM-ATR proteins play a crucial role in detecting DNA damage and activating downstream signaling pathways that result in the activation of P53, which can induce cell cycle arrest or apoptosis. M checkpoint, spindle assembly checkpoint. The M checkpoint ensures that all chromosomes are properly aligned and attached to the meiotic spindle before anaphase begins. MAD2 and BUBR1 proteins are involved in sensing whether the chromosomes are properly aligned at the metaphase plate. If any chromosomes are not correctly attached to spindle fibers, the checkpoint prevents the cell from progressing into anaphase until all chromosomes are aligned correctly. The anaphase-promoting complex, cyclosome APC-C, when activated, triggers the degradation of securin and cyclins, leading to the separation of sister chromatids and progression of the cell cycle. Tumor suppressors and oncogenes in cell cycle regulation. The cell cycle is tightly controlled by both tumor suppressors and oncogenes. Tumor suppressors inhibit cell cycle progression, while oncogenes promote it. Mutations in these regulators can lead to uncontrolled cell division and contribute to cancer development. Tumor suppressors. P53, known as the guardian of the genome, P53 plays a critical role in monitoring DNA integrity. If DNA damage is detected, P53 induces cell cycle arrest in the G1, G2, or M phase to allow repair. If the damage is irreparable, P53 can trigger apoptosis. RB, retinoblastoma protein, RB controls the G1 to S phase transition by binding to and inhibiting E2F transcription factors. Inactivation of RB, e.g., through mutations leads to the unregulated progression of the cell cycle. P21, this cyclin-dependent kinase inhibitor is activated by P53 in response to DNA damage. P21 inhibits CDK activity, thereby causing cell cycle arrest and allowing time for DNA repair. Oncogenes. Cyclin D1, overexpression of cyclin D1 can lead to premature progression through the G1 checkpoint, bypassing regulatory controls and promoting unregulated cell division. CDK4/6, mutations or overexpression of CDK4 or CDK6 can lead to unregulated phosphorylation of RB, driving the cell cycle forward even in the presence of DNA damage or incomplete DNA replication. E2F, overactivation of E2F transcription factors, due to mutations in RB, leads to the expression of genes required for DNA replication, driving cell cycle progression. Regulation of the cell cycle by external signals. External signals, such as growth factors, hormones, and nutrients, can also regulate the cell cycle. These signals bind to cell surface receptors and trigger intracellular signaling pathways that regulate cyclin CDK activity and other cell cycle regulators. Growth factors, these proteins stimulate cells to enter the cell cycle by promoting the synthesis of cyclins and the activation of CDKs. Epidermal growth factor, EGF, is one example of a growth factor that binds to cell surface receptors, activating the ROS signaling pathway, which eventually activates cyclin DCDK4 complexes. Nutrient availability, the availability of nutrients, such as amino acids and glucose, regulates the cell cycle by influencing the activity of cyclin CDK complexes. In conditions of nutrient deprivation, cells may halt the cycle and enter a resting state, G0. Clinical relevance. Understanding the regulation of the cell cycle is crucial for cancer research and therapy. Aberrant regulation, caused by mutations in genes encoding cyclins, CDKs, or tumor suppressors, can lead to the development of cancer. Targeting these cell cycle regulators with specific inhibitors has become a promising strategy in cancer treatment. Solved previous year question. Q. Which of the following proteins is involved in the G1 to S phase transition by releasing E2F transcription factors? A. P53B, cyclin DC, RBD, MAD2 answer, C. RB. Explanation. The RB retinoblastoma protein binds to E2F transcription factors and inhibits the expression of genes required for DNA synthesis. When RB is phosphorylated by cyclin D CDK4 complexes, it releases E2F, allowing the cell to proceed from G1 to S phase. MCQs. Which of the following proteins is responsible for the progression of the cell cycle from G1 to S phase by phosphorylating RB? A. Cyclin DB, cyclin EC, P53D, cyclin A. Which checkpoint ensures that DNA replication is complete and that no DNA damage is present before the cell enters mitosis? A. G1 checkpoint B, S checkpoint C, G2 checkpoint D, M checkpoint. Which of the following proteins is activated by DNA damage to induce cell cycle arrest? A. Cyclin AB, P53C, RBD, E2F. Let me know if you'd like to explore another topic or continue with more questions. You said. Programmed cell death, aging and senescence. ChatGPT said. Programmed cell death, aging, and senescence. Programmed cell death (PCD), aging, and cellular senescence are interrelated biological processes that play essential roles in the development, maintenance, and decline of an organism. While PCD is a natural process crucial for tissue homeostasis and organismal health, aging and senescence involve the gradual deterioration of cellular functions, contributing to the aging process and the onset of age-related diseases. Programmed cell death (PCD). Programmed cell death refers to a regulated process by which cells undergo self-destruction in response to various internal or external signals. Unlike necrosis, which is uncontrolled and results from injury or disease, PCD is a controlled process essential for maintaining the balance between cell proliferation and cell death in tissues. PCD is critical in processes such as development, e.g., the sculpting of fingers and toes during embryogenesis, immune system regulation, e.g., the elimination of self-reactive immune cells, and maintaining tissue homeostasis. There are two major forms of programmed cell death, apoptosis and autophagy. 1. Apoptosis. Apoptosis is a highly regulated form of programmed cell death that occurs in a series of well-defined steps, involving signaling pathways, gene expression changes, and the activation of specific proteases. Intrinsic pathway. This pathway is triggered by intracellular stress signals, such as DNA damage, oxidative stress, or mitochondrial dysfunction. It involves the release of cytochrome C from the mitochondria, which activates caspases, cysteine aspartic proteases. Activated caspases then cleave a variety of cellular substrates, leading to the characteristic morphological changes seen in apoptosis, such as membrane blebbing, chromatin condensation, and cell fragmentation into apoptotic bodies, which are phagocytosed by surrounding cells. Extrinsic pathway. This pathway is triggered by external signals such as death ligands, e.g., FASL or TNF-alpha binding to the respective receptors on the cell surface, leading to the activation of caspases through the death receptor pathway. This pathway is important in immune responses and elimination of infected or transformed cells. Key regulators. BCL2 family proteins. The BCL2 family plays a central role in regulating apoptosis. Pro-apoptotic members, e.g., BAC promote mitochondrial outer membrane permeabilization, MOMP, leading to cytochrome C release, while anti-apoptotic members, e.g., BCL2, BCLXL, inhibit this process. Caspases. These proteases are central to apoptosis execution. Caspase 3 is one of the most important executioner caspases, leading to the cleavage of vital cellular proteins. 2. Autophagy. Autophagy is another form of programmed cell death that is primarily involved in cellular homeostasis and quality control. It is characterized by the degradation and recycling of damaged organelles, misfolded proteins, and other cellular components. Mechanism. During autophagy, cytoplasmic material is sequestered into vesicles called autophagosomes, which then fuse with lysosomes for degradation. This process is mediated by a variety of proteins, such as ATG, autophagy-related genes, and is essential for maintaining cellular health, especially during stress conditions such as nutrient deprivation. Role in cell death. While autophagy can act as a survival mechanism under stress, excessive or dysregulated autophagy can contribute to cell death. In some instances, autophagy and apoptosis can work together to ensure appropriate cell removal in pathological conditions. Aging. Aging is the process of gradual decline in cellular and tissue function, leading to the diminished ability of an organism to maintain homeostasis and respond to environmental stress. The aging process is complex and involves genetic, environmental, and lifestyle factors that contribute to cellular damage over time. Cellular mechanisms of aging, DNA damage and mutations. Over time, cells accumulate DNA damage due to internal factors, e.g., replication errors, reactive oxygen species, and external factors, e.g., UV radiation, toxins. The accumulation of DNA mutations impairs the ability of cells to function properly, and inefficient DNA repair mechanisms exacerbate this process. Telomere shortening. Telomeres are repetitive DNA sequences at the ends of chromosomes that protect them from degradation and prevent fusion with other chromosomes. With each cell division, telomeres shorten, eventually leading to cellular senescence or cell death when they reach a critical length. Telomere shortening is thought to be one of the key drivers of aging at the cellular level. Mitochondrial dysfunction. Mitochondria, the powerhouses of the cell, accumulate damage over time due to oxidative stress and mutations in mitochondrial DNA. This leads to impaired ATP production and the generation of more reactive oxygen species, ROS, which in turn damage cellular components and exacerbate the aging process. Epigenetic changes. Aging is also associated with alterations in the epigenome, such as changes in DNA methylation, histone modifications, and chromatin remodeling. These changes can lead to the deregulation of gene expression, contributing to cellular dysfunction and aging. Protein misfolding and aggregation. Over time, the ability of cells to maintain protein homeostasis, proteostasis declines, leading to the accumulation of misfolded proteins and protein aggregates. These aggregates can impair cellular function and contribute to age-related diseases such as Alzheimer's and Parkinson's. Cellular senescence. Cellular senescence is a state of irreversible cell cycle arrest that occurs in response to various stressors, including DNA damage, telomere shortening, oncogene activation, or oxidative stress. Senescent cells no longer divide, but they remain metabolically active and secrete a variety of pro-inflammatory factors known as the senescence-associated secretory phenotype, SASP. Mechanisms of senescence. DNA damage response, DDR. In response to DNA damage or stress, cells activate the p53p21 pathway, which induces cell cycle arrest, allowing time for DNA repair. If the damage is too severe or cannot be repaired, the cell enters senescence. Telomere-induced senescence. As telomeres shorten with each cell division, they eventually reach a critical length, triggering a DNA damage response that leads to senescence. This is often referred to as replicative senescence. Oncogene-induced senescence. Oncogenes, when overexpressed, can induce premature senescence as a protective mechanism to prevent uncontrolled cell division. This type of senescence is called oncogene-induced senescence, OIS. Senescence-associated secretory phenotype, SASP. Senescent cells secrete a variety of factors, including pro-inflammatory cytokines, growth factors, and proteases. While the SASP can promote tissue repair, chronic accumulation of senescent cells and their secretions contributes to aging and age-related diseases by creating a pro-inflammatory environment. Role of senescence in aging. While senescence is a protective mechanism that prevents the proliferation of damaged cells, the accumulation of senescent cells over time can contribute to aging and age-related diseases. The inflammaging associated with the SASP can damage surrounding tissues, impairing tissue regeneration and contributing to diseases such as osteoarthritis, cardiovascular disease, and neurodegenerative disorders. Clinical relevance of PCD, aging, and senescence. Understanding the molecular mechanisms underlying programmed cell death, aging, and senescence has significant implications for aging research and age-related diseases. Therapeutic strategies aimed at modulating these processes, such as senolytic drugs which selectively eliminate senescent cells, or telomerase activation to delay telomere shortening, hold promise for extending lifespan and treating age-related diseases. Solved previous year question. Q. Which of the following is a key hallmark of programmed cell death? Apoptosis. A. Chromatin condensation. B. Membrane rupture. C. Mitochondrial fusion. D. Increased cell volume. Answer. A. Chromatin condensation. Explanation. Apoptosis is characterized by several morphological features, including chromatin condensation, membrane blebbing, and the fragmentation of the cell into apoptotic bodies. Membrane rupture and cell swelling are more indicative of necrosis. MCQs. Which of the following factors is primarily responsible for inducing cellular senescence due to DNA damage? A. Telomere shortening. B. P53 activation. C. Increased proteasomal activity. D. Mitochondrial fusion. What is the primary function of a senescence-associated secretory phenotype? SASP. A. Promoting cell cycle progression. B. Inducing inflammation and tissue repair. C. Stimulating apoptosis. D. Enhancing DNA repair. Which of the following processes is most associated with aging at the cellular level? A. Telomere extension. B. Increased DNA repair capacity. C. Accumulation of oxidative damage. D. Increased mitochondrial function. Let me know if you'd like to continue with further topics. 2 halves. You said. Give answers to those MCQ from now on all responses. ChatGPT said. Certainly. Below are the answers to the MCQs from the previous response. MCQs answers. Which of the following factors is primarily responsible for inducing cellular senescence due to DNA damage? Answer. B. P53 activation. Explanation. The activation of P53 in response to DNA damage leads to the induction of cell cycle arrest, which can ultimately result in cellular senescence if the damage is irreparable. What is the primary function of a senescence-associated secretory phenotype? SASP. Answer. B. Inducing inflammation and tissue repair. Explanation. The SASP involves the secretion of pro-inflammatory cytokines, growth factors, and proteases, which promote tissue repair but also contribute to the chronic inflammation seen in aging and age-related diseases. Which of the following processes is most associated with aging at the cellular level? Answer. C. Accumulation of oxidative damage. Explanation. Oxidative damage from reactive oxygen species ROS over time is a key contributor to cellular aging, leading to damage of DNA, proteins, and lipids, and impairing cellular functions. Let me know if you have more topics or need further explanations. You said. Cell communication and signaling, general principles of cell communication. ChatGPT said. Cell communication and signaling, general principles of cell communication. Cell communication is a fundamental aspect of cellular life, ensuring that cells in a multicellular organism can coordinate their functions to maintain homeostasis, development, and responses to environmental stimuli. This communication occurs through a variety of signaling mechanisms and pathways, enabling cells to transmit information to one another and respond to changes in their internal and external environments. Cell communication can be divided into 2 broad categories: intracellular communication, where cells communicate within themselves, and intercellular communication, where signals are transmitted between cells. These signaling pathways are crucial for processes such as growth, differentiation, immune response, and tissue repair. Key principles of cell communication. Signal reception. The first step in cell communication is the reception of a signaling molecule, ligand, by a receptor. This interaction is highly specific, with receptors recognizing and binding to particular signaling molecules. Receptors are typically proteins located either on the cell surface or inside the cell, depending on the nature of the signaling molecule. Cell surface receptors. These are typically membrane-bound proteins that respond to hydrophilic ligands, e.g., peptides, growth factors, neurotransmitters. The ligands bind to these receptors, initiating intracellular signaling cascades. Examples of these receptors include G-protein coupled receptors, GPCRs, receptor tyrosine kinases, RTKs, and ion channel receptors. Intracellular receptors. These receptors are located in the cytoplasm or nucleus and are usually activated by hydrophobic signaling molecules, such as steroid hormones, thyroid hormones, and retinoids. When these molecules bind, they often result in the activation of transcription factors, leading to changes in gene expression. Signal transduction. Once a receptor is activated by its ligand, the signal must be transduced into the cell, meaning the extracellular signal is converted into an intracellular response. This process often involves a cascade of molecular events that amplify the signal and transmit it to different parts of the cell. Second messengers. Many signaling pathways involve the generation of small molecules known as second messengers, which help propagate the signal inside the cell. Common second messengers include cyclic AMP, GAMP, calcium ions, Ca2, and inositol phosphate, IP. These molecules amplify the signal and can activate various downstream effectors, such as protein kinases. Kinase signaling cascades. Protein kinases, which add phosphate groups to target proteins, are key players in signal transduction. For example, the MAPK, mitogen-activated protein kinase, pathway is a major signaling cascade involved in cell proliferation, differentiation, and survival. Phosphatases and GPases. These molecules also play important roles in regulating signal transduction by removing phosphate groups, phosphatases, or hydrolyzing GTP to GDP, GTPases, turning off certain pathways. Signal integration and amplification. Signals often converge and interact with other signaling pathways, leading to integrated cellular responses. The amplification of a signal ensures that a small amount of the ligand can elicit a large cellular response. This is typically achieved through multiple enzyme activations, second messenger production, and feedback loops that increase or decrease the signaling output. Crosstalk between pathways. Different signaling pathways can interact with each other, either synergistically or antagonistically. For instance, the PI3K ACT pathway can modulate the response of the MAPK pathway, affecting cell survival and growth. Feedback loops. Feedback mechanisms, both positive and negative, are essential for regulating the strength and duration of signaling. For example, a negative feedback loop may inhibit the activation of a signaling pathway after it reaches a certain threshold, while positive feedback can amplify the response and sustain it longer. Signal response. The final stage in cell communication is the cellular response to the signal, which can vary depending on the type of signal, the receptor, and the cell involved. Responses can be classified into short-term and long-term effects. Short-term responses. These are often rapid and reversible, such as changes in enzyme activity, ion channel function, or cytoskeletal rearrangements. For example, signaling pathways controlling muscle contraction or neurotransmitter release can produce immediate effects. Long-term responses. These typically involve changes in gene expression, leading to the synthesis of new proteins and alteration of cellular functions. Long-term responses are critical for processes like cell growth, differentiation, and adaptation to changes in the environment. Termination of signaling. For cell communication to remain regulated and precise, signaling must be turned off once the appropriate response has been achieved. Various mechanisms exist to terminate signaling, including the degradation of second messengers, dephosphorylation of proteins by phosphatases, internalization or degradation of receptors, and the hydrolysis of GTP by GTPases. Receptor desensitization and internalization. Some receptors, especially GPCRs, undergo desensitization after prolonged ligand binding, which reduces their responsiveness to further signals. This can be achieved through phosphorylation by specific kinases, leading to receptor internalization and degradation or recycling. Types of cell signaling. Autocrine signaling. In autocrine signaling, the signaling molecules are released by the cell and bind to receptors on the same cell, or other cells of the same type. This type of signaling plays a crucial role in the regulation of processes such as immune cell activation and tumor cell proliferation. Paracrine signaling. In paracrine signaling, signaling molecules are secreted by a cell and affect nearby cells in the local environment. This type of signaling is common in the nervous system, where neurotransmitters transmit signals between adjacent neurons, and in the regulation of inflammation. Endocrine signaling. Endocrine signaling involves the release of hormones into the bloodstream, where they travel long distances to target cells in distant tissues or organs. Examples of hormones involved in endocrine signaling include insulin, thyroid hormones, and steroids. Juxtacrine signaling. In juxtacrine signaling, cells communicate through direct contact. This often occurs through membrane-bound signaling molecules that bind to receptors on an adjacent cell. An example of juxtacrine signaling is the interaction between Notch receptors and ligands during developmental processes. Examples of signaling pathways. G-protein coupled receptor (GPCR) pathway. GPCRs are a large family of receptors involved in sensing a variety of extracellular signals such as light, neurotransmitters, and hormones. Upon ligand binding, these receptors activate G-proteins, which in turn regulate the production of second messengers like GAMP and IP. GPCRs mediate a wide range of physiological responses, including vision, immune responses, and neurotransmission. Receptor tyrosine kinase (RTK) pathway. RTKs are receptors for many growth factors and hormones. When the ligand binds to an RTK, it triggers receptor dimerization and autophosphorylation of tyrosine residues in the intracellular domain. This phosphorylation recruits adapter proteins and activates downstream signaling pathways such as the MAPK pathway, which regulates cell growth and differentiation. WNT signaling pathway. The WNT signaling pathway is critical in regulating embryonic development, tissue homeostasis, and cancer. In the absence of WNT ligands, a protein complex prevents the activation of beta-catenin. Upon WNT binding, the complex is inactivated, leading to the accumulation of beta-catenin, which enters the nucleus and activates the transcription of target genes involved in cell proliferation and differentiation. Solved previous year question. Q. Which of the following best describes the mechanism of action of G-protein coupled receptors (GPCRs)? A. GPCRs activate second messengers through receptor dimerization. B. GPCRs directly phosphorylate target proteins. C. GPCRs activate G-proteins, which in turn stimulate second messengers. D. GPCRs mediate gene transcription directly. Answer. C. GPCRs activate G-proteins, which in turn stimulate second messengers. Explanation. GPCRs transmit extracellular signals by activating G-proteins, which then regulate the production of second messengers such as GAMP, IP, or DAG, which propagate the signal inside the cell. MCQs. Which of the following best describes a characteristic feature of endocrine signaling? A. Short-range signaling within a localized area. B. Hormones travel through the bloodstream to distant target cells. C. The signaling molecules act on the same cell. D. Direct cell-to-cell contact is required. What is the role of second messengers in signal transduction? A. To amplify the signal inside the cell. B. To degrade the signaling molecule. C. To bind to cell surface receptors. D. To maintain the cell membrane potential. In which type of signaling do cells communicate by direct contact through membrane-bound molecules? A. Autocrine signaling. B. Endocrine signaling. C. Juxtacrine signaling. D. Paracrine signaling. MCQs. Answers. Answer. B. Hormones travel through the bloodstream to distant target cells. Answer. A. To amplify the signal inside the cell. Answer. C. Juxtacrine signaling. Let me know if you'd like further clarifications or more topics. You said, cell. Adhesion and roles of different adhesion molecules. ChatGPT said. Cell adhesion and roles of different adhesion molecules. Cell adhesion is a crucial process that enables cells to interact with one another and with the extracellular matrix, ECM. These interactions are essential for maintaining the integrity of tissues, allowing for proper cell function, communication, and the formation of organized structures. Adhesion molecules are specialized proteins that mediate these interactions and can be classified into various categories based on their structure, location, and function. Cell adhesion plays a fundamental role in processes like tissue morphogenesis, wound healing, immune responses, and cancer metastasis. The mechanisms of cell adhesion are largely governed by the interaction between cell adhesion molecules, GAMs, integrins, and components of the extracellular matrix. Key types of adhesion molecules. Cadherins. Cadherins are a family of glycoproteins that mediate calcium-dependent cell adhesion. They are crucial for the establishment of adherens junctions and desmosomes, which contribute to the structural integrity and mechanical stability of tissues. Cadherins play a key role in cell sorting during embryonic development and tissue formation. Classical cadherins. These include E. cadherin, epithelial cells, N. cadherin, neural cells, and P. cadherin, placenta. These cadherins bind to similar cadherins on adjacent cells, ensuring that cells of the same type adhere to one another. Desmosomal cadherins. These include desmogleins and desmoglins, which are components of desmosomes, providing mechanical strength to tissues subjected to stress, such as cardiac and epithelial tissues. Function. Cadherins mediate the formation of strong cell-cell junctions, facilitating tissue architecture and integrity. Their role in embryogenesis includes promoting cellular differentiation, polarity, and organization. In tissues like the skin and heart, cadherins help withstand mechanical forces. Integrins. Integrins are transmembrane receptors that facilitate cell extracellular matrix, ECM, adhesion. They are involved in the physical connection between the intracellular cytoskeleton and the ECM, which includes fibronectin, collagen, laminin, and other matrix components. The physical connection between the intracellular cytoskeleton and the ECM, which includes fibronectin, collagen, laminin, and other matrix components. Structure. Integrins are heterotomeric proteins, consisting of alpha and beta subunits. Different combinations of these subunits allow integrins to interact with distinct ECM proteins, leading to diverse functions in cell adhesion, migration, and signal transduction. Examples. Alpha 5 beta 1 integrin binds to fibronectin, while alpha 6 beta 4 integrin interacts with laminin. These interactions are vital for processes such as cell migration during wound healing, tissue development, and immune responses. Function: integrins regulate the interaction between cells in the ECM, mediating cellular responses to mechanical signals and promoting cell survival, differentiation, and migration. In processes like wound healing, integrins assist in the migration of epithelial cells to close wounds. Selectins. Selectins are a group of adhesion molecules that mediate cell-cell interactions during inflammation and immune responses. They are primarily involved in the transient adhesion between leukocytes, white blood cells, and endothelial cells of blood vessels. Types of selectins. There are three major types of selectins: E-selectin endothelial cells, P-selectin platelets and endothelial cells, L-selectin leukocytes. Mechanism: selectins bind to specific carbohydrate structures on glycoproteins present on the surface of other cells, promoting rolling interactions of leukocytes along the blood vessel wall. This interaction is a critical early step in the inflammatory response, allowing immune cells to migrate to sites of infection or injury. Function: selectins are critical for the early stages of immune cell trafficking and for the recruitment of leukocytes to sites of inflammation. They also play a role in the adhesion of platelets to endothelial cells during clot formation. Immunoglobulin superfamily CAMs, IGSF CAMs. These molecules are a diverse group of cell surface proteins involved in cell-cell adhesion, particularly in immune responses and neuronal development. The IGSF includes a variety of adhesion molecules, such as ICAMs, intracellular adhesion molecules, VCAMs, vascular cell adhesion molecules, and NCAMs, neural cell adhesion molecules. ICAMs and VCAMs. These molecules are expressed on endothelial cells and interact with integrins on leukocytes. This interaction is essential for the extravasation of immune cells from the bloodstream into tissues during inflammatory responses. NCAMs, found in the nervous system. NCAMs mediate adhesion between neurons, contributing to processes such as synapse formation, neuronal migration, and axon guidance. Function: IGSF CAMs are involved in immune cell adhesion and migration, neuronal development, and the formation of tight junctions in endothelial cells during inflammation. They contribute to processes like tissue repair, immune surveillance, and synaptic plasticity. Tight junctions TJ and adherens junctions AJ. Although these are not adhesion molecules per se, they involve proteins that help maintain the physical integrity of tissues by facilitating adhesion between adjacent cells. Tight junctions TJ. These junctions are formed by proteins such as claudins and occludins, which create a selective barrier to the diffusion of molecules across epithelial and endothelial layers. They are crucial for maintaining the polarity of cells and for controlling the paracellular transport of ions and solutes. Adherens junctions AJ. These junctions involve E-cadherin and link the actin cytoskeletons of adjacent cells. They play a key role in maintaining cell shape and the integrity of tissue architecture. Function: tight junctions prevent leakage of solutes between cells, while adherens junctions help in maintaining tissue structure and resisting mechanical stress. MCQs. Which of the following is a characteristic of cadherens in cell adhesion? A. They are involved in cell matrix adhesion. B. They are calcium dependent. C. They mediate short-range signaling between cells. D. They are primarily involved in immune responses. Which of the following integrins binds to fibronectin? A. Alpha-1 beta-1 integrin. B. Alpha-3 beta-1 integrin. C. Alpha-5 beta-1 integrin. D. Alpha-6 beta-4 integrin. What is the role of selectins in immune responses? A. They mediate long-term adhesion between immune cells. B. They promote the rolling of leukocytes along the endothelial cell surface. C. They are involved in the adhesion of platelets to blood vessels. D. They mediate the migration of immune cells across the blood-brain barrier. MCQs answers. Answer: B. They are calcium dependent. Answer: C. Alpha-5 beta-1 integrin. Answer: B. They promote the rolling of leukocytes along the endothelial cell surface. This concludes the detailed explanation of cell adhesion and the roles of different adhesion molecules. Let me know if you'd like further clarifications or additional topics. You said. Gap junctions, extracellular matrix. ChatGPT said. Gap junctions. Gap junctions are specialized intercellular connections that enable direct communication between adjacent cells. They consist of protein channels called connexons, which allow the passage of small molecules and ions between cells, facilitating coordinated cellular responses. These junctions are critical in tissues where rapid and synchronized activity between cells is necessary, such as in muscle contraction, neural signaling, and hormone secretion. Structure of gap junctions. Connexins. Gap junctions are formed by hexameric assemblies of proteins called connexins, which are members of a large family of transmembrane proteins. Each connexin molecule spans the plasma membrane 4 times and has both intracellular and extracellular loops. The channels formed by connexins are called connexons. Connexon formation. Each connexon is composed of 6 connexin subunits that align in a head-to-head configuration, forming a channel between adjacent cells. When 2 connexons from neighboring cells align, they create a continuous channel that allows the exchange of ions, small metabolites, and signaling molecules. Regulation. The opening and closing of gap junctions are regulated by factors such as calcium ion concentration, pH, and voltage changes. This regulation ensures that gap junctions are open when communication between cells is required and closed when necessary to maintain cellular homeostasis. Function of gap junctions. Intercellular communication. Gap junctions allow for the direct passage of ions, nucleotides, second messengers e.g., CAMP, and other small molecules between cells. This direct transfer of information enables cells to synchronize their activities, such as the coordinated contraction of cardiac and smooth muscle cells or the synchronized firing of neurons. Homeostasis and coordination. In tissues like the heart, gap junctions ensure the synchronized contraction of myocardial cells by allowing the rapid transmission of electrical signals. In the nervous system, gap junctions can facilitate communication between neurons for the propagation of action potentials. Regulation of tissue function. Gap junctions are involved in maintaining tissue homeostasis by allowing the exchange of metabolic and signaling molecules between cells. For example, they help coordinate the movement of ions and nutrients in epithelial and endothelial layers, contributing to the proper functioning of organs and tissues. Extracellular matrix (ECM). The extracellular matrix (ECM) is a complex network of macromolecules that provides structural and biochemical support to surrounding cells. It plays a crucial role in regulating cell behavior, tissue development, and maintaining tissue integrity. The ECM is composed of proteins, polysaccharides, and various other molecules that interact with the plasma membrane of cells. Components of the ECM: fibrous proteins. Collagen. Collagen is the most abundant protein in the ECM and provides tensile strength to tissues. It forms fibrous structures that resist stretching and are found in connective tissues, skin, bone, cartilage, and blood vessels. Elastin. Elastin is a highly elastic protein that allows tissues to return to their original shape after stretching or deformation. It is abundant in tissues such as the lungs, skin, and arteries, which require elasticity for proper function. Fibronectin. Fibronectin is a glycoprotein that helps anchor cells to the ECM and plays a role in cell migration, wound healing, and tissue repair. It also binds to integrins, connecting the ECM to the intracellular cytoskeleton. Proteoglycans. Proteoglycans are large molecules composed of a core protein with one or more glycosaminoglycan (GAG) chains attached. These molecules have a high capacity to attract and retain water, contributing to the hydration and turgor of tissues. They are involved in providing structural support, modulating signaling pathways, and maintaining the integrity of the ECM. Glycosaminoglycans (GAGs). GAGs are long, unbranched polysaccharide chains that are often attached to a core protein to form proteoglycans. Examples of GAGs include hyaluronic acid, chondroitin sulfate, and heparin sulfate. GAGs contribute to the ECM's ability to retain water and form a gel-like structure that resists compression forces, particularly in cartilaginous tissues. Hyaluronic acid. Hyaluronic acid is a non-sulfated GAG that exists as a free polysaccharide or is incorporated into proteoglycans. It is important for tissue hydration, wound healing, and cell migration. Its large size and ability to bind water make it essential in tissues that need to maintain high moisture levels. Laminins. Laminins are glycoproteins found in the basal lamina of the ECM, where they play a significant role in cell adhesion, differentiation, migration, and signaling. Laminins interact with integrins on the cell surface and help organize the ECM by interacting with collagen and other ECM components. Functions of the ECM. Structural support. The ECM provides mechanical strength and structural integrity to tissues, ensuring that they maintain their shape and resist deformation under stress. Collagen fibers provide tensile strength, while elastin fibers allow tissues to stretch and return to their original form. Cellular support. The ECM acts as a scaffold, anchoring cells in place and providing them with a supportive microenvironment. It regulates cell behavior, including cell adhesion, migration, differentiation, and proliferation. Cell signaling. The ECM plays a crucial role in transmitting mechanical and biochemical signals to cells. Components like fibronectin, laminin, and proteoglycans interact with cell surface receptors, e.g., integrins, to regulate signaling pathways that influence cellular responses such as survival, growth, and differentiation. Wound healing. The ECM facilitates tissue repair and regeneration by guiding cell migration and proliferation during wound healing. It provides a scaffold for the formation of new tissues and helps in the remodeling process. Tissue development and morphogenesis. During embryonic development and tissue formation, the ECM regulates cell migration, differentiation, and patterning. It helps establish the tissue architecture and plays a vital role in processes like angiogenesis and organogenesis. MCQs. Which of the following is a primary component of gap junctions? A. Collagen. B. Connexin. C. Laminins. D. Elastin. Which ECM component is responsible for providing tensile strength to tissues? A. Hyaluronic acid. B. Collagen. C. Elastin. D. Fibronectin. Which of the following ECM components is involved in regulating cell signaling and cell migration? A. Collagen. B. Laminin. C. Elastin. D. Proteoglycans. MCQs. Answers. Answer. B. Connexins. Answer. B. Collagen. Answer. B. Laminins. This concludes the detailed explanation of gap junctions in the extracellular matrix. If you have any further questions or need more topics covered, feel free to ask. You said. Integrins, neurotransmission and its regulation, hormones and the receptors, ChatGPT said. Integrins. Integrins are a family of transmembrane receptors that mediate the attachment of a cell to its surrounding extracellular matrix, ECM, and to other cells. They play a crucial role in various cellular processes such as adhesion, migration, differentiation, survival, and signal transduction. Integrins are heterodimers, composed of alpha and beta subunits, which determine the specific ECM proteins that the integrin will bind to. These receptors are involved in cellular interactions with proteins like fibronectin, collagen, laminin, and vitronectin, which are important components of the ECM. Structure of integrins. Heterodimeric structure. Integrins are composed of two subunits, one alpha-alpha subunit and one beta-beta subunit. Each subunit consists of extracellular, transmembrane, and intracellular domains. The extracellular domain binds to ECM proteins, while the intracellular domain interacts with the cytoskeleton, mainly actin filaments. Subunit variability. There are at least 18 different alpha subunits and 8 beta subunits, leading to the formation of at least 24 unique integrin combinations, each of which binds to distinct ECM ligands. Ligand binding. Integrins bind to their ligands through their extracellular domains. The most common ECM components bound by integrins are fibronectin, collagen, laminin, and vitronectin. Function of integrins. Cell ECM adhesion. Integrins mediate the attachment of cells to the ECM, which is crucial for maintaining the structural integrity of tissues. This adhesion supports cell survival and provides the mechanical linkage between the cytoskeleton and the ECM. Signal transduction. Integrins are involved in signal transduction pathways. When integrins bind to ECM proteins, they activate intracellular signaling pathways that regulate cell migration, proliferation, and survival. This process is often referred to as outside-in signaling. Cell migration. Integrins play a vital role in cell migration, which is necessary for processes such as wound healing, immune responses, and embryogenesis. Integrins form focal adhesions that connect the cytoskeleton to the ECM, allowing cells to crawl across surfaces. Regulation of gene expression. Integrin-mediated signaling can also influence gene expression through pathways like the focal adhesion kinase FAK pathway, Rho-GTPases, and the MAPK-ERK pathway, which regulate various cellular responses, including differentiation and cell cycle progression. Neurotransmission and its regulation. Neurotransmission is the process by which neurons communicate with each other and with other cell types, e.g., muscle cells, gland cells, using chemical signals called neurotransmitters. This communication is essential for the functioning of the nervous system and regulates various physiological processes such as movement, cognition, emotion, and autonomic function. Mechanism of neurotransmission. Synthesis and storage of neurotransmitters. Neurotransmitters are synthesized in the presynaptic neuron from precursors and stored in vesicles within the synaptic terminal. Release of neurotransmitters. When an action potential reaches the synaptic terminal, it triggers the opening of voltage-gated calcium channels. The influx of calcium ions causes synaptic vesicles to fuse with the presynaptic membrane and release neurotransmitters into the synaptic cleft. Receptor binding. The neurotransmitters diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane. These receptors are typically classified as ionotropic receptors, which directly control ion channels, or metabotropic receptors, which activate intracellular signaling pathways via G proteins. Termination of signal. After neurotransmitters bind to the receptors, the signal is terminated by several mechanisms. Reuptake. Neurotransmitters are taken back into the presynaptic neuron for recycling. Enzymatic degradation. Some neurotransmitters are broken down by enzymes in the synaptic cleft. Diffusion. Neurotransmitters can also diffuse away from the synaptic cleft. Regulation of neurotransmission. Presynaptic regulation. Neurotransmitter release can be modulated by factors such as autoregulation, where neurotransmitter release is controlled by receptors on the presynaptic neuron, feedback from postsynaptic receptors, and external factors like drugs and hormones. Receptor sensitivity. The sensitivity of postsynaptic receptors can be altered through upregulation or downregulation, which influences the strength of neurotransmission. Neurotransmitter modulation. The release and effects of neurotransmitters can be influenced by various substances, such as neuromodulators which modify the release of neurotransmitters, drugs, and toxins. For example, serotonin and dopamine can modulate the release of other neurotransmitters and affect mood and behavior. Hormones and their receptors. Hormones are chemical messengers that are produced by specialized glands and travel through the bloodstream to regulate various physiological processes in distant target cells. Hormones can be broadly classified into peptides, steroids, and amines, and they act by binding to specific receptors located on the surface of target cells or within the cytoplasmic nucleus. Types of hormone receptors. Cell surface receptors. These receptors are typically proteins located on the cell membrane and bind to hydrophilic hormones, e.g., peptides, catecholamines. The receptor binding leads to the activation of intracellular signaling pathways that result in a cellular response. G-protein coupled receptors, GPCRs, these are the most common type of surface receptors. Upon hormone binding, they activate G-proteins, which in turn activate or inhibit various intracellular signaling pathways, e.g., CAMP, phosphatidylinositol turnover. Receptor tyrosine kinases, RTKs. These receptors have intrinsic kinase activity and, upon ligand binding, they phosphorylate themselves in downstream proteins, leading to activation of signaling pathways that regulate growth, differentiation, and metabolism. Intracellular receptors. These receptors are found in the cytoplasm or nucleus and are activated by lipophilic hormones, e.g., steroid hormones, thyroid hormones. Once the hormone binds, the receptor undergoes a conformational change and can directly influence gene transcription. Steroid hormone receptors. Steroid hormones, such as estrogen and testosterone, diffuse through the cell membrane and bind to intracellular receptors, which then bind to specific DNA sequences and regulate gene expression. Thyroid hormone receptors. Thyroid hormones bind to intracellular receptors in the nucleus, modulating the transcription of genes involved in metabolism, growth, and development. Hormone action. Signal transduction. When a hormone binds to its receptor, it triggers a cascade of intracellular signaling events that modulate the activity of enzymes, ion channels, or transcription factors within the target cell. Regulation of cellular processes. Hormones regulate various processes such as growth, metabolism, immune responses, and reproductive functions. For example, insulin regulates glucose uptake in cells, while thyroid hormones regulate basal metabolic rate. Feedback mechanisms. Hormonal systems are often regulated by feedback loops, where the action of a hormone is regulated by its own effect. For example, the hypothalamus and pituitary gland regulate the secretion of thyroid hormones through a negative feedback mechanism. MCQs. Which of the following is a characteristic feature of integrins? A. They form ion channels. B. They mediate cell-to-cell adhesion. C. They are involved in cell migration and ECM interaction. D. They are only present in epithelial. C. Calcium influx to the presynaptic neuron. D. Chloride influx into the presynaptic neuron. Which of the following hormones acts via intracellular receptors? A. Insulin. B. Epinephrine. C. Estrogen. D. Glucagon. MCQs. Answers. Answer. C. They are involved in cell migration and ECM interaction. Answer. C. Calcium influx into the presynaptic neuron. Answer. C. Estrogen. This concludes the detailed explanation of integrins, neurotransmission, and hormones and their receptors. Let me know if you'd like further elaboration or additional topics. You said, cell surface. Receptor, signaling through G-protein coupled receptors, ChatGPT said. Cell surface receptors. Cell surface receptors are specialized proteins located on the outer surface of the plasma membrane that bind to signaling molecules, ligands such as hormones, neurotransmitters, and growth factors. These receptors act as transducers, converting the extracellular signal into an intracellular response. Surface receptors are essential in mediating a variety of physiological processes including immune responses, neurotransmission, and cell differentiation. There are several classes of cell surface receptors, each with a distinct mechanism of action. The most important types include G-protein coupled receptors, GPCRs, receptor tyrosine kinases, RTKs, ion channel link receptors, and the most important types include G-protein coupled receptors, GPCRs, receptor tyrosine kinases, RTKs, ion channel link receptors, and integrins. Among these, GPCRs are the most ubiquitous and versatile. G-protein coupled receptors, GPCRs. GPCRs are the largest family of cell surface receptors and are involved in transmitting signals from extracellular molecules to the inside of the cell. They are integral membrane proteins characterized by their ability to couple with G-proteins to transmit signals across the cell membrane. These receptors are involved in a wide range of physiological processes, such as sensory perception, vision, taste, and smell, neurotransmission, and immune responses. Structure of GPCRs. 7-transmembrane alpha. 7-transmembrane GPCRs have a structure consisting of 7 membrane-spanning alpha helices that traverse the plasma membrane. These helices form a pocket that binds the ligand, typically on the extracellular side. Extracellular ligand binding domain. The N-terminal region of the receptor is located extracellularly, and it is responsible for ligand recognition and binding. Intracellular G-protein binding domain. The C-terminal region of the receptor is located intracellularly and is responsible for interacting with the associated G-protein. G-protein interaction site. The receptor undergoes a conformational change upon ligand binding, allowing it to interact with a G-protein complex on the cytoplasmic side of the membrane. Mechanism of signaling through GPCRs. Ligand binding. The process begins when a specific ligand, such as a hormone, neurotransmitter, or sensory molecule, binds to the extracellular domain of the GPCR. This binding induces a conformational change in the receptor structure. G-protein activation. The conformational change in the GPCR enables it to interact with an associated G-protein in the cytoplasmic membrane. G-proteins are heterotrimers, composed of 3 subunits, alpha, alpha, beta, beta, and gamma, gamma. The alpha subunit binds guanine nucleotides, GTP or GDP. GDP-GTP exchange. When the GPCR activates the G-protein, it catalyzes the exchange of GDP for GTP on the alpha subunit. This exchange activates the G-protein by causing the alpha subunit to dissociate from the beta and gamma subunits. Signal transduction via G-protein subunits. The activated G-protein subunits, alpha GTP and BG, can interact with various intracellular effectors. These effectors can be enzymes, ion channels, or other signaling proteins. Common downstream effectors include adenylyl cyclase AC. The alpha subunit, especially in G's proteins, can activate adenylyl cyclase, which catalyzes the conversion of ATP to cyclic AMP, CAMP. CAMP acts as a second messenger that activates protein kinase A, pKa. Phospholipase C, PLC. Some G-protein subunits, e.g., GQ, activate PLC, which catalyzes the breakdown of phosphatidylinositol bisphosphate, BIP2, into inositol trisphosphate, IP3, and diacylglycerol DAG. IP3 increases intracellular calcium levels, while DAG activates protein kinase C (PKC). Termination of signal. The GTP bound to the alpha subunit is hydrolyzed to GDP by the intrinsic GTPase activity of the alpha subunit. This hydrolysis leads to the reassociation of the alpha subunit with the BG subunits, thus terminating the signal and resetting the G protein cycle. Additionally, other mechanisms such as receptor desensitization and endocytosis help terminate the signal. Types of G proteins. G's proteins. These stimulate adenylyl cyclase, leading to increased CAMP levels. GI proteins. These inhibit adenylyl cyclase, decreasing CAMP levels, and can also activate phospholipase CGQ proteins. These activate phospholipase C to generate IP3 and DAG. G12-13 proteins. These are involved in regulating the actin cytoskeleton and cell movement. Biological significance of GPCRs. GPCRs mediate a broad range of biological responses that are vital for cellular function and organismal homeostasis. Neurotransmission. GPCRs mediate signaling in the nervous system by responding to neurotransmitters like dopamine, serotonin, and norepinephrine. Sensory perception. GPCRs play a key role in the senses, including vision through opsins, taste, and smell. Cardiovascular function. GPCRs regulate heart rate and blood pressure by modulating neurotransmitter release and smooth muscle contraction. Immune response. GPCRs are involved in immune signaling pathways, including chemotaxis of immune cells to sites of infection. Endocrine regulation. GPCRs are critical for hormone signaling, including the regulation of insulin, thyroid hormones, and adrenaline. MCQs. Which of the following is a key feature of G protein coupled receptors (GPCRs)? A. They have a single transmembrane domain. B. They activate G proteins upon ligand binding. C. They are only involved in ion channel opening. D. They are located exclusively in the nucleus. What is the role of GTP in G protein signaling? A. It activates the G protein by replacing GDP. B. It deactivates the G protein by replacing GTP. C. It serves as the second messenger in the signaling pathway. D. It binds to the beta subunit of the G protein. Which of the following is the most common second messenger produced by G protein coupled receptors? A. Inositol trisphosphate IP3B, cyclic AMP, CAMP, C. Calcium ions, Ca², D. Diacylglycerol, DAG, MCQs. Answers. Answer. B. They activate G proteins upon ligand binding. Answer. It activates the G protein by replacing GDP. Answer. B. Cyclic AMP, CAMP. This concludes the detailed explanation of cell surface receptors, specifically focusing on G protein coupled receptors (GPCRs). If you need further clarification or have more topics to cover, feel free to let me know. You said. Signal transduction pathways. Second messengers. ChatGPT said. Signal transduction pathways. Signal transduction refers to the process by which a cell converts an extracellular signal into a functional cellular response. This complex cascade of events involves receptors, second messengers, protein kinases, and other intracellular signaling molecules. Signal transduction is essential for the regulation of a variety of cellular functions, such as gene expression, cell growth, differentiation, metabolism, and apoptosis. The process begins when an extracellular signal ligand, such as a hormone, neurotransmitter, or growth factor, binds to a receptor on the cell surface or inside the cell. The receptor then transmits the signal across the membrane or within the cell, often involving a series of protein-protein interactions and the activation of second messengers. Types of signaling pathways. Receptor tyrosine kinase (RTK) mediated signaling. This pathway is activated by the binding of ligands like growth factors to receptor tyrosine kinases (RTKs). Upon ligand binding, RTKs undergo dimerization and autophosphorylation, which activates downstream signaling proteins. Key pathway. MAPK mitogen-activated protein kinase pathway, which regulates cell division, differentiation, and survival. G protein coupled receptor (GPCR) mediated signaling. As previously discussed, GPCRs activate G proteins, which then modulate the activity of effector enzymes e.g., adenylyl cyclase, phospholipase C, or ion channels. This leads to the production of second messengers, such as CAMP, IP3, and DAG. This pathway is involved in various physiological processes, including neurotransmission, immune responses, and sensory perception. Ion channel mediated signaling. Certain receptors, like ligand-gated ion channels, open in response to ligand binding, allowing ions e.g., Ca² not to flow into the cell, leading to changes in membrane potential and activation of downstream signaling pathways. This is particularly important in neurotransmission and muscle contraction. Intracellular receptor-mediated signaling. Some ligands, such as steroid hormones, pass through the plasma membrane and bind to intracellular receptors in the cytoplasm or nucleus. These receptors, often nuclear hormone receptors, act as transcription factors that regulate gene expression. Example, corticosteroid and thyroid hormone signaling. Second messengers in signal transduction. Second messengers are small, intracellular molecules that are generated in response to the activation of a receptor by ligand. These messengers amplify the signal inside the cell and help mediate the cell's response to the external stimulus. Common second messengers include CAMP, calcium ions, Ca², inositol trisphosphate, IP3, and diacylglycerol DAG. Types of second messengers. Cyclic AMP CAMP. CAMP is produced from ATP by adenylyl cyclase, which is activated by G protein coupled receptors (GPCRs). Once produced, CAMP activates protein kinase A (pKa) by binding to its regulatory subunits and releasing the catalytic subunits, which can then phosphorylate target proteins. Functions of CAMP. Regulation of metabolic processes, e.g., glycogen breakdown in the liver. Control of gene expression through the activation of pKa and subsequent activation of transcription factors like CREB. Modulation of heart rate and smooth muscle relaxation. Calcium ions, Ca². Ca² acts as a second messenger in many signaling pathways. It is stored in organelles such as the endoplasmic reticulum and mitochondria, and released into the cytoplasm in response to various signals. Key mechanisms for the release of Ca² include IP3 and ryanodine receptors on the ER. Functions of Ca². Regulates processes like muscle contraction, neurotransmitter release, and gene expression. Activates various calcium-binding proteins, such as calmodulin, which in turn activates other enzymes, e.g., calmodulin-dependent kinase. Inositol trisphosphate, IP3. IP3 is generated from PIP2 phosphatidylinositol bisphosphate by the action of phospholipase C (PLC). IP3 then binds to receptors on the endoplasmic reticulum, causing the release of Ca² from intracellular stores. Functions of IP3. Elevates cytosolic calcium levels, which regulates numerous cellular processes. Works in concert with DAG to activate protein kinase C (PKC), another important signaling enzyme. Diacylglycerol DAG. DAG is produced along with IP3 from the hydrolysis of PIP2 by PLC. While IP3 mediates the release of Ca² from the air, DAG stays in the plasma membrane and activates protein kinase C (PKC). Functions of DAG. Activates PKC, which then phosphorylates various proteins involved in cell growth, differentiation, and survival. Nitric oxide, NO. NO is a unique second messenger because it is a gaseous molecule. It is synthesized from arginine by nitric oxide synthase (NOS) in response to signaling pathways. NO activates guanylate cyclase, leading to the production of cGMP, a second messenger that regulates vascular tone and smooth muscle relaxation. Functions of NO. Regulates blood flow by causing vasodilation. Modulates neurotransmission and immune responses. Cyclic GMP, cGMP. cGMP is produced by guanylate cyclase in response to NO or other signals. It activates protein kinase G (PKG), which then phosphorylates proteins involved in cell growth, metabolism, and muscle relaxation. Functions of cGMP. Mediates vasodilation in response to nitric oxide. Involved in regulating ion channel activity and phototransduction in the retina. MCQs. Which of the following is the second messenger involved in the activation of protein kinase A (PKA)? A. Inositol-trisphosphate IP3 diacylglycerol DAG. C. Cyclic AMP GAMP. D. Calcium ions Ca². What is the primary function of inositol-trisphosphate IP3 in signal transduction? A. Activation of protein kinase C (PKC). B. Release of calcium ions from the endoplasmic reticulum. C. Conversion of ATP to cyclic AMP. D. Activation of guanylate cyclase. Which second messenger is directly involved in vasodilation through the activation of protein kinase G (PKG)? A. Cyclic GMP cGMP. B. Cyclic AMP GAMP. C. Diacylglycerol DAG. D. Nitric oxide. No. MCQs. Answers. Answer. C. Cyclic AMP GAMP. Answer. B. Release of calcium ions from the endoplasmic reticulum. Answer. A. Cyclic GMP cGMP. This concludes the explanation of signal transduction pathways and second messengers. If you need further clarification or have more topics to cover, feel free to ask. You said. Regulation of signaling pathways, bacterial and plant two-component systems, light signaling in plants, bacterial chemotaxis and quorum sensing, ChatGPT said. Regulation of signaling pathways. Signaling pathways must be precisely regulated to ensure proper cellular responses to external stimuli. The regulation of signaling involves several mechanisms to modulate the intensity, duration, and specificity of a signal. This regulation is crucial for maintaining cellular homeostasis, coordinating complex biological processes, and preventing pathological conditions such as cancer or autoimmune diseases. Key regulatory mechanisms include receptor desensitization and downregulation. Upon prolonged exposure to a ligand, receptors may undergo desensitization, which reduces their responsiveness. This is often achieved through phosphorylation of the receptor by kinases, e.g., G-protein coupled receptor kinases, GPCRKs, leading to the binding of beta-arrestins, which prevent further signaling. Receptor downregulation involves receptor internalization and degradation. This is particularly important in pathways involving growth factors or hormones, where constant activation could lead to pathological cell growth. Negative feedback loops. Signaling pathways often involve negative feedback mechanisms, where the output of a pathway inhibits the upstream signaling events. For example, the activation of MAPK, mitogen-activated protein kinase, cascades can lead to the expression of proteins that inhibit the signaling pathway at an early stage. Phosphatases. Phosphorylation and dephosphorylation of proteins are central to the regulation of most signaling pathways. Protein phosphatases, e.g., protein tyrosine phosphatases, remove phosphate groups from proteins, reversing the effects of kinases and thus terminating the signal. Crosstalk between pathways. Signaling pathways often interact with one another in a process known as crosstalk. For instance, the activation of CAMP-dependent pathways can modulate the activity of MAPK pathways. Crosstalk ensures that the cell integrates multiple signals to produce a coordinated response. Proteolytic cleavage. Some signaling molecules, such as transcription factors or proteins involved in apoptosis, are regulated by proteolytic cleavage. For example, the Notch signaling pathway is regulated by proteases that cleave the Notch receptor, enabling it to enter the nucleus and regulate gene expression. Bacterial and plant two-component systems. Two-component systems (TCS) are signaling mechanisms used by both bacteria and plants to sense and respond to environmental changes. They are composed of two main components, a sensor kinase and a response regulator. Bacterial two-component system. In bacteria, the sensor kinase is typically a histidine kinase, which detects specific environmental signals, e.g., changes in temperature, pH, or the presence of specific molecules. Upon activation, the sensor kinase autophosphorylates itself on a histidine residue. The phosphate group is then transferred to a response regulator, often a transcription factor, which modulates gene expression to initiate the appropriate cellular response. The two-component system is essential for processes such as bacterial chemotaxis, virulence, antibiotic resistance, and sporulation. Plant two-component system. In plants, two-component systems are involved in regulating various processes, such as growth, development, stress response, and hormone signaling. Similar to bacteria, plant systems include a sensor kinase that detects environmental signals and a response regulator that activates downstream responses. The main difference is that plant response regulators are typically transcription factors that regulate gene expression in response to the signal. For example, the ethylene signaling pathway in plants involves a two-component system that helps regulate responses to stress, fruit ripening, and senescence. Light signaling in plants. Light is a critical environmental cue for plants, influencing processes such as photomorphogenesis, phototropism, flowering, and photosynthesis. Plants use a variety of light receptors, primarily phytochromes, cryptochromes, and phototropines, to perceive light and initiate signaling cascades. Phytochromes. Phytochromes are light-sensitive proteins that perceive red and far-red light. Upon absorption of red light, phytochromes undergo a conformational change and become active, promoting gene expression that controls processes such as seed germination, shade avoidance, and flowering. The transition between the active and inactive forms of phytochromes allows plants to detect light changes and adjust their growth accordingly. Cryptochromes and phototropins. Cryptochromes are blue light receptors involved in regulating processes like the circadian clock, flowering, and seedling development. Phototropins are blue light receptors that play a key role in phototropism, growth toward light, and stomatal opening, allowing plants to optimize light capture for photosynthesis. Signaling pathways in response to light. Upon light absorption by these receptors, various signaling pathways are activated, including the activation of transcription factors that regulate gene expression. These pathways influence the expression of genes involved in cell elongation, chlorophyll synthesis, and flowering time. Bacterial chemotaxis and quorum sensing. Bacterial chemotaxis. Chemotaxis refers to the ability of bacteria to move toward favorable conditions, positive chemotaxis, or away from harmful substances, negative chemotaxis. This process is regulated by two-component systems and involves sensory receptors that detect chemical gradients in the environment. The key components of chemotaxis include methyl-accepting chemotaxis proteins, MCPs. These receptors detect changes in the concentration of attractants, e.g., sugars, amino acids, or repellents, e.g., toxins. The signal from MCPs is transduced to J proteins, which modulate the activity of the flagella motor to control bacterial movement. This process helps bacteria find optimal environments for growth and survival. Quorum sensing. Quorum sensing is a mechanism of cell-to-cell communication in bacteria that allows them to coordinate their behavior based on population density. Bacteria release signaling molecules called autoinducers. When the concentration of autoinducers reaches a threshold, it triggers a collective response in the population, such as biofilm formation, virulence factor production, or antibiotic resistance. Examples of quorum sensing systems include Al-N-acyl homozerine lactones in gram-negative bacteria, such as Vibrio fischeri bioluminescence and Pseudomonas aeruginosa biofilm formation. Peptides in gram-positive bacteria, such as Streptococcus pneumoniae, antibiotic resistance. MCQs. Which component of the two-component system in bacteria is responsible for detecting environmental signals? A. Response regulator. B. Histidine kinase. C. Phosphatase. D. Transcription factor. Which light receptor in plants primarily mediates responses to red and far-red light? A. Cryptochromes. B. Phytochromes. C. Phototropins. D. Rhodopsins. Which of the following processes is primarily regulated by bacterial quorum sensing? A. Photosynthesis. B. Chemotaxis. C. Biofilm formation. D. Germination. MCQs. Answers. Answer. B. Histidine kinase. Answer. B. Phytochromes. Answer. C. Biofilm formation. This concludes the discussion on signaling pathway regulation, two-component systems, light signaling in plants, bacterial chemotaxis, and quorum sensing. Feel free to reach out for more topics or clarifications. You said, Toll-like receptors. ChatGPT said, Toll-like receptors. TLRs. Toll-like receptors. TLRs are a family of pattern recognition receptors, PRRs, that play a critical role in the innate immune system, which is the body's first line of defense against pathogens. These receptors are involved in detecting pathogen-associated molecular patterns, PAMPs, and damage-associated molecular patterns, DAMPs, both of which are indicators of infection or tissue damage. TLRs are expressed on various immune cells, such as macrophages, dendritic cells, and neutrophils, and they initiate signaling pathways that lead to immune responses. Structure of Toll-like receptors. TLRs are transmembrane proteins that typically consist of an extracellular domain, a single transmembrane domain, and an intracellular domain. The extracellular domain is responsible for recognizing and binding to specific patterns found on pathogens or damaged cells. The intracellular domain contains a Toll-L1 receptor TIR domain, which is essential for initiating intracellular signaling pathways once the receptor is activated. There are 10 different types of TLRs in humans, TLR1 through TLR10, each recognizing distinct PAMPs or DAMPs. For instance, TLR1 recognizes triacylipopeptides found in bacterial membranes. TLR2 binds peptidoglycans, lipoteichoic acid, and other bacterial components. TLR3 recognizes double-stranded RNA associated with viral infections. TLR4 detects lipopolysaccharides LPS from gram-negative bacteria. TLR5 recognizes flagellin, a protein component of bacterial flagella. TLR9 binds unmethylated CpG DNA, which is commonly found in bacterial and viral genomes. Mechanism of action. Upon binding to their ligands, TLRs undergo a conformational change that triggers the activation of intracellular signaling pathways. This signaling usually involves the recruitment of adapter proteins such as MYD88, myeloid differentiation factor 88, and TRIF-TIR domain-containing adapter-inducing interferon beta. These adapter proteins activate downstream signaling cascades that result in the activation of transcription factors like NF-kappa B, nuclear factor kappa B, and IRFs, interferon regulatory factors, which are critical for the production of pro-inflammatory cytokines, interferons, and other immune molecules. MYD88 dependent signaling pathway. This pathway is activated by most TLRs except TLR3. After binding, MYD88 is recruited to the TIR domain of TLRs, leading to the activation of IRAKs, IL-1 receptor-associated kinases, which then activate TOC1 transforming growth factor beta-activated kinase 1, and IKKs, I-kappa B kinases. These proteins ultimately activate NF-kappa B, a transcription factor that promotes the expression of pro-inflammatory cytokines like TNF-alpha, IL-1, and IL-6. TRIF-dependent signaling pathway. This pathway is specifically activated by TLR3 and TLR4. Upon ligand binding, TRIF is recruited to the TLRs, initiating signaling that leads to the activation of IRF3, which drives the production of type 1 interferons such as IF and beta. These interferons are key players in antiviral immunity. Role in immune response. TLRs play a fundamental role in innate immunity by recognizing and responding to pathogens early in infection. The immune response initiated by TLR activation leads to the production of cytokines that coordinate the response of other immune cells, such as macrophages, neutrophils, T cells, and B cells. Some of the main functions of TLRs in immune response include detection of pathogens. TLRs recognize PAMPs, which are highly conserved structures found in pathogens, such as bacteria, viruses, fungi, and parasites. This enables the host to detect a broad range of infections. Inflammatory response. Upon activation, TLRs trigger the release of pro-inflammatory cytokines like IL-6, TNF-alpha, and IL-1 beta, which promote inflammation and recruit immune cells to the site of infection. This inflammatory response is crucial for clearing infections but must be tightly regulated to prevent tissue damage. Activation of adaptive immunity. In addition to activating the innate immune system, TLRs also promote the activation of adaptive immunity. For example, TLR signaling enhances the function of dendritic cells, which are essential for presenting antigens to T cells and initiating adaptive immune responses. Tissue repair and homeostasis. TLRs also play roles in tissue repair and maintaining immune homeostasis. Their activation in response to DAMPs released from damaged tissues helps coordinate the resolution of inflammation and tissue healing. TLRs and disease. While TLRs are essential for defending the body against infections, their dysregulation can lead to a variety of diseases, including autoimmune diseases. Overactivation of TLRs can lead to excessive inflammation and contribute to autoimmune diseases such as rheumatoid arthritis, systemic lupus erythematosus (SLE), and inflammatory bowel disease (IBD). Chronic inflammatory diseases. Inappropriate or persistent activation of TLRs may lead to chronic inflammatory conditions, which are associated with diseases like atherosclerosis, diabetes, and cancer. Sepsis. Overactivation of TLRs during bacterial infections can result in a hyperinflammatory response, leading to sepsis, a life-threatening condition characterized by widespread inflammation and organ failure. Cancer. TLRs have a dual role in cancer, where their activation can both promote tumor progression and enhance anti-tumor immunity. In some cases, TLRs can stimulate the immune system to recognize and destroy cancer cells, while in other cases, chronic TLR activation can promote the growth and metastasis of tumors. Conclusion. Toll-like receptors are critical for detecting pathogens and initiating the innate immune response. Their ability to sense PAMPs and DAMPs allows for rapid defense mechanisms that shape both the innate and adaptive immune responses. However, improper regulation of TLR signaling can result in a variety of diseases, making them an important target for therapeutic interventions in inflammatory, autoimmune, and infectious diseases. MCQs. The following TLRs recognizes lipopolysaccharide, LPS, from gram-negative bacteria. A. TLR1B, TLR3C, TLR4D, TLR9. Which of the following is not a result of toll-like receptor activation? A. Production of pro-inflammatory cytokines. B. Activation of NF-kappa-B. Suppression of immune responses. D. Induction of interferon production. Which of the following TLRs recognizes double-stranded RNA from viral infections? A. TLR1B, TLR3C, TLR4D, TLR7. MCQs. Answers. Answer. C. TLR4. Answer. C. Suppression of immune responses. Answer. B. TLR3. This concludes the detailed explanation on toll-like receptors, TLRs, and their role in immunity. Let me know if you need more topics. You said. Ligand-gated ion channel receptors and integrins, signaling through intracellular receptors. ChatGPT said. Ligand-gated ion channel receptors and integrins. Ligand-gated ion channel receptors. Ligand-gated ion channel receptors are a class of membrane-bound proteins that mediate the rapid transmission of signals across the plasma membrane by directly controlling the flow of ions in response to the binding of specific ligands. These receptors are crucial for mediating fast synaptic transmission in neurons and muscle cells, and they also play a role in other physiological processes such as hormone signaling, sensory perception, and muscle contraction. Structure. Ligand-gated ion channels are typically composed of multiple subunits that form a central ion channel core. These receptors are pentameric or tetrameric in structure, depending on the type of receptor. The binding of a ligand such as a neurotransmitter causes a conformational change in the receptor that opens or closes the ion channel, allowing ions such as Na+, K+, Ca2+, or Cl to flow through the channel. This ion flux results in changes in the membrane potential and initiates cellular responses. Mechanism of action. The mechanism of action of ligand-gated ion channels is as follows. A ligand, for example, acetylcholine, glutamate, or GABA binds to the extracellular domain of the receptor. This binding induces a conformational change in the receptor that opens the ion channel. Ions, such as Na+, K+, Ca2+, or Cl, flow through the channel down their concentration gradient. The influx or efflux of ions alters the membrane potential of the cell, leading to various downstream effects. For example, the influx of Na+ can lead to depolarization of the membrane, which may trigger action potentials in neurons or muscle cells. Examples. Common examples of ligand-gated ion channels include nicotinic acetylcholine receptor, NACHR, a receptor found at the neuromuscular junction that opens a channel permeable to Na+ and K+ when acetylcholine binds to it, leading to muscle contraction. Ionotropic glutamate receptors, AMPA, NMDA, found in the brain. These receptors mediate excitatory neurotransmission by allowing Na+ and Ca2+ influx upon glutamate binding, leading to synaptic plasticity. GABA receptors. When the neurotransmitter GABA binds to the receptor, it opens a chloride ion channel, GABA receptors. When the neurotransmitter GABA binds to the receptor, it opens a chloride ion channel, resulting in hyperpolarization of the membrane and inhibition of neural activity. Integrins. Integrins are a family of transmembrane receptors that mediate cell extracellular matrix (ECM) interactions and play a critical role in various cellular processes such as adhesion, migration, differentiation, and signal transduction. They are composed of alpha and beta subunits, which form heterodimeric receptors that interact with ligands in the ECM, such as fibronectin, collagen, and laminin. Structure. Integrins are heterodimeric receptors, meaning that each receptor is composed of two subunits, an alpha subunit and a beta subunit. These subunits are non-covalently associated and together form a receptor with a large extracellular ligand binding domain, a single transmembrane helix, and a short intracellular tail. The extracellular domain binds to ECM components while the intracellular domain interacts with the cytoskeleton through adaptor proteins. Mechanism of action. Integrins function by converting mechanical signals from the ECM into biochemical signals within the cell in a process known as outside-in signaling. The mechanism involves the following steps. The extracellular domain of the integrin binds to ECM ligands such as fibronectin or collagen. The binding induces a conformational change in the integrin, which leads to the recruitment of intracellular signaling proteins. These signaling proteins, such as focal adhesion kinase (FAK), SRC, and paxillin, interact with the cytoskeleton, regulating cell shape, motility, and adhesion to the matrix. Integrins also participate in inside-out signaling, where intracellular signals modulate the integrin affinity for ECM ligands. This is crucial for regulating cell adhesion and migration. Functions. Cell adhesion. Integrins mediate the adhesion of cells to the ECM, which is crucial for tissue integrity, development, and wound healing. Cell migration. Integrins are involved in cell movement by linking the cytoskeleton to the ECM, allowing cells to crawl along substrates. Signal transduction. Integrins transmit mechanical signals from the ECM to intracellular signaling pathways that regulate cell behavior, including growth, differentiation, and survival. Immune response. Integrins are involved in leukocyte trafficking, enabling immune cells to migrate to sites of infection or inflammation. Signaling through intracellular receptors. Intracellular receptors. Intracellular receptors are a class of receptors located within the cytoplasm or nucleus of the cell. These receptors bind lipophilic hydrophobic ligands such as steroid hormones, thyroid hormones, and retinoids, which can easily diffuse across the lipid bilayer of the plasma membrane. Once inside the cell, these ligands bind to their corresponding receptors, leading to the activation of specific signaling pathways that typically result in changes to gene expression. Structure. Intracellular receptors can be classified into two major groups based on their location. Cytoplasmic receptors. These receptors are located in the cytoplasm and are typically cytosolic until they bind their ligand. Upon ligand binding, they undergo a conformational change that allows them to translocate to the nucleus. Nuclear receptors. These receptors are located in the nucleus and are bound to specific DNA sequences in the promoter regions of target genes. Mechanism of action. The general mechanism of intracellular receptor signaling follows these steps. Ligand binding. The ligand, such as a steroid hormone, diffuses through the plasma membrane and binds to its intracellular receptor in the cytoplasm or nucleus. Receptor activation. Upon ligand binding, the receptor undergoes a conformational change that either exposes or creates a DNA binding domain, allowing the receptor ligand complex to bind to specific sequences of DNA known as hormone response elements (HREs). Transcriptional regulation. The receptor ligand complex acts as a transcription factor, regulating the expression of specific target genes by recruiting CO activators or corepressors to the promoter region. Physiological response. The change in gene expression leads to various physiological effects, such as the regulation of metabolism, immune responses, growth, and differentiation.